students, welcome to Biomix. Do you know how different plants survive in different temperature? There are certain plants like maize, sorghum, sugarcane. These plants can survive when the temperature is high. But in the same temperature or in the high temperature, the rice cannot survive. Why it is so? It is due to certain modifications in the cells of the plants which are present in high temperature. So in this video, we will discuss about the C4 cycle or the Hatch sex pathway which are been adapted by the plants which are present in high temperature. So what happens in the plants for the fixation of the carbon? There are certain cycles like it is C3 cycle or C4 cycle. In case of the C3 cycle, the carbon dioxide fixation takes place in the mesophyll cell. So why it is known as C3 cycle? Because the first stable compound which is produced here is a 3 carbon containing compound. And in the C4 plants, the first stable compound which is produced, it is a 4 carbon containing compound. So what is the difference between the C3 plants and the C4 plants? There are certain modifications here. Here the carbon dioxide fixation in the C3 plants takes place in the mesophyll cell. But in the C4 plants, the carbon dioxide fixation takes place in the bundled sheath shell. Okay, so the carbon dioxide fixation takes place by a enzyme which is known as rubisco, and this enzyme is having two activity. That is, it can also work as a carboxylase as well as oxygenase. Okay, so here when the temperature is high, in case of high temperature, this rubisco work as a oxygenase. It works as a oxygenase, due to which photorespiration takes place. So in case of photorespiration, what happens? The oxygen is taken in by the plant and it releases carbon dioxide, which is a wasteful process. But here in case of C3 cycle, when the temperature is low and the amount of CO2 is high, here this enzyme works as a carboxylase. So here there is no problem, but here there is a problem. So in the plant there is being modification of the cell. Here the mesophyll cell is produced and it forms a sheath-like structure. After crossing this only, it will enter into the bundled sheath shell, and here the temperature in this, the temperature is low. It is low as compared to the upper part. Here the temperature will be low and the carbon dioxide will be accumulated here, and here the fixation of the carbon will take place by this rubisco enzyme. Here the enzyme is present in the C4 plants. The rubisco enzyme is present in the bundled sheath cell. Okay, but in case of C3 plant, the rubisco enzyme which is present, it is present in the mesophyll cell, and here let's see how this process is taking place. So here the carbon dioxide which is present in the environment, it is first transferred into the mesophyll cell. Then after some modification or after some changes, this carbon which is present, it is being transferred into the bundled sheath cell, and here the Calvin cycle or the C3 cycle is taking place in presence of the light. Okay, let us understand how this process is taking place in details. So in the atmosphere, carbon dioxide is present and it has been taken into the mesophyll cell. After the transfer, this carbon dioxide is being converted into bicarbonate in presence of the enzyme carbonic anhydrase. Okay, and it requires H2O and it requires water. So the first compound which can accept this carbon in the form of bicarbonate is the phosphoenol pyruvate. Okay, and this phosphoenol, and after receiving this in presence of one of the enzyme which is known as PEP carboxylase, it is being converted into a 4 carbon containing compound which is known as oxaloacetate. This phosphoenol pyruvate, what it was? It was a 3 carbon compound, and after accepting this carbon dioxide in presence of the enzyme PEP carboxylase, it has been converted into 4 carbon containing compound which is oxaloacetate. Okay, after this, by the utilization of NADPH to NADP plus in presence of the enzyme malate dehydrogenase, this oxaloacetate is converted into another 4 carbon containing compound which is known as malate. This is a process which is taking place in the mesophyll cell. After this malate is being transferred into the bundled sheath cell through the help of plasmodesmata. From plasmodesmata, it has been transferred into the bundled sheath cell. Now, in presence of malic enzyme, this is being converted into pyruvate. This pyruvate, it is a 3 carbon containing compound. 1 carbon is being lost over here, and during this process, this NADH in the mesophyll cell, what happens? The NADPH was utilized and it produced the NADP plus. This NADP plus is being utilized in the bundle sheath shell for the formation of NADPH. Okay, so here there was utilization of the NADPH, here there is gain of NADPH. So here there is no more utilization, or in net there is no utilization of NADPH. NADP is present, NADPH is present over here. So during this process there is loss of 1 carbon dioxide. This carbon dioxide is being stored in the bundle sheath shell, and this carbon dioxide is undergoing Calvin cycle or C3 cycle in the bundle sheath cell. Okay, so here the enzyme which is present, phosphoenol pyruvate, it cannot accept the oxygen. Okay, if the concentration of oxygen is high in the environment also, this enzyme cannot take the oxygenase. It has a high affinity for bicarbonate, HCO3 minus bicarbonate. Then it is having the affinity with CO2, but it is having no affinity with oxygenase. That is the reason it cannot bind with this oxygenase. Okay, so here there is release of the carbon dioxide in the bundle sheath cell, and here the Calvin cycle is taking place. In this way, the modification has been done in the plant which can survive in the high temperature. So what happens? Here the Rubisco enzyme was creating some problem. So what they did, they changed the place of the Rubisco enzyme for doing or fixing the carbon dioxide in the form of Calvin cycle. Okay. So now here there is formation of pyruvate. This pyruvate is again transferred into the mesophyll cell for recycling of this 3 carbon compound which is known as phosphoenol pyruvate. Okay, and here the pyruvate is present. And in presence of pyruvate phosphate dikinase, it is being converted into phosphoenol pyruvate, and during this process ATP is being utilized. Here the ATP is giving the AMP. So here 2-phosphate is being released. So if it is using ATP and it is giving AMP, that means it is using by default 2 ATP. It is equivalent to utilization of 2 ATP. So for fixation of 1 carbon dioxide, it requires 2 extra ATP molecule. So if it is utilizing 2 ATP extra, why it is utilizing 2 ATP in extra? Because if it will not change its position for carbon dioxide fixation, here the Rubisco enzyme, if it is present in the mesophyll cell, during this time it can work as a oxygenase. Okay, so here photorespiration will take place. So as compared to photorespiration, utilizing 2 ATP and fixation of carbon in the bundle sheath cell is more appropriate, or it requires less energy. Okay, I hope this video is helpful for you, and if you are having any doubt in this video regarding this C4 cycle, please do ask me in the comment section. Thank you everyone for watching this video.